Hello, fellow neuroscience enthusiasts. This video is another installment of the Brain Preservation Foundation's Neuroscience Journal Club, in which we briefly review the results of a selected paper and discuss its relevance to the ongoing debate regarding brain preservation. Today's paper describes a detailed biophysical model of mouse primary visual cortex. According to Google Scholar, it has been cited 111 times since its publication in 2020. It is entitled Systematic Integration of Structural and Functional Data into Multiscale Models of Mouse Primary Visual Cortex. Please note that although I will try my best to accurately relate the research, it is always possible that I may misinterpret something crucial. If I do, please let me know in the comments and I will try to correct it. Another thing to note is that this is a more detailed, complex version of a 2018 paper by many of the same authors. That paper described a model of cortical layer 4. This model extends that to include 5 cortical layers. Let's start with a quick overview. The study features a detailed biophysical model of mouse primary visual cortex consisting of 230,000 neurons arranged in 5 cortical layers. Connectivity between the neurons was generated by sampling statistical distributions, which closely follow known empirical data and present equivalent computational challenges to when connectivity obtained from anatomical electron microscopy reconstructions is used. The model can be run using arbitrary visual stimuli as input, which you see on the left of this slide, and produces population-level activity patterns, which you see on the right side. Thereby, it helps to elucidate structure-function relationships and the decoding of function from purely anatomical data. The model was constructed in a data-driven fashion from what is known about the biological circuit organization, following the dictum attributed to Richard Feynman, "What I cannot create, I do not understand." We understand the phenomena that we observe experimentally to the extent that we can reproduce them. The focus on mouse V1 is due to the substantial amounts of high-quality data being available, and the primary aim is to provide a computational platform to study cortical structure and computation in general under realistic biological constraints. This model can also be used as templates for other cortical areas. 2 variants of the model were created, a version using point neuron models or single compartment models, and a full version using detailed compartmental morphology containing hundreds of compartments per cell. The left panel here pertains to the full model. It used 112 unique neuron models with variations in biophysics and morphology. The authors instantiate then an 845 millimeter radius of cortex, which you see illustrated on the bottom right. For the biophysically detailed variant, the core, or the 400 millimeter radius center, is composed of these multi-compartmental neurons with somatic Hodgkin-Huxley dynamics and passive dendrites. And these are surrounded by an annulus of simple single compartmental leaky integrating fire neurons to avoid boundary artifacts. This is illustrated in the top right figure, which is actually from the paper preceding this one, the 2018 paper. Although recent surveys suggest 50 to 100 cell classes in V1, the currently available neuronal models, connectivity data, and in vivo recordings limit the range to 17 classes, illustrated in the left panel here. These 17 cell classes are represented by 112 unique individual models by combining them with morphological data. Note that neuron positions are defined in physical space. Each of these neurons has an X, Y, and Z coordinate, whereas visual stimuli supplied to the model, as well as the LGN filters, are defined in visual space. Thus, a mapping between physical space and visual space needs to be defined. The geometric center of the neuronal population corresponds to the center of visual space, and the mapping around it was then created based on known data on retinotomy. Visual degrees traversed per millimeter of cortex are 70 degrees per millimeter in the azimuth and 40 degrees per millimeter in elevation. Note the substantial asymmetry between these 2 directions. This will turn out to be important later. LGN model first then creates action potentials for arbitrary visual stimuli and feeding it to the V1 model. 17,400 LGN units cover visual space. This is actually quite close to the estimated 18,000 neurons in mouse LGN, but note the absence of a distinct core shell region in this model. Each unit looks at a certain part of the visual space defined by a Gaussian spatial kernel or receptive field, and it has a biphasic temporal kernel illustrated on the bottom right. Several different on and off type response kernels are defined in the model. The firing rate output of the LGN is then converted via Poisson processes into spike times, and synaptic strengths from LGN to V1 were adjusted on the basis of trying to obtain an empirically constrained synaptic current measured using a full field grading stimulus to match experimental conditions. Every V1 neuron was assigned a preferred temporal frequency and preferred orientation. These are just drawn from a distribution with means as measured experimentally. These are distributed in a non-periodic fashion, generating the so-called salt and pepper type of cortical map as observed also empirically in mouse V1. Then the connectivity is generated to obtain these desired functional responses. In addition to orientation selectivity, direction selectivity is a prominent characteristic of V1 neurons. How it is generated is a central question in the field. The mechanism employed here is perhaps surprisingly simple. Direction selectivity is known to be produced in V1 from convergence of spatiotemporally asymmetric LGN inputs. The authors assume that LGN innovation into V1 neurons has two subfields, one with a slow or sustained kinetics and the other with a fast or transient kinetics. These produce an asymmetry in responses to opposite directions of motion, as you can see in the right panel here. The authors targeted LGN inputs to V1 neurons, so the placement of these subfields, using the following procedure. For every V1 neuron, they determined the visual center size and directionality of elliptical subfields by sampling these parameters from experimentally constrained distributions. The authors then identified LGN receptive fields that overlap with these elliptical subfields of the V1 neuron, one subfield always sampling from transient LGN filters and the other subfield from a sustained filter. Then the strength of synapses is established to match the target mean current in response to a drifting grading. Layer 4 is the primary input target of the LGN, and therefore the current amplitudes are largest in this layer. You can see the largest spiking response here in the right panel. To test the outcome of this procedure, the authors carried out simulations of the entire V1 network, but without recurrent connections, using drifting grading stimuli, and indeed individual neurons were found to be direction selective using this connectivity scheme, which you can see an example of in the bottom left. That was the feed-forward or bottom-up connectivity. Now to determine the proper recurrent connectivity in the cortical model, V1 circuits feature extensive recurrent connections which amplify LGN inputs and shape V1 computation. The assumption was made of an 85% excitatory and 15% inhibitory fraction of neurons. No resources exist that comprehensively characterize all connections under standardized conditions. So a collection of various literature sources was used from other parts of mouse cortex as well as rat cortex. 89 of the total 289 entries remained undetermined. These are the empty cells in the top left panels and were set to 0 due to lack of data. Using the data, estimates were formulated of connection probability, synaptic amplitude, or the strength of synapses, these two you see in the top left, the axonal delays of connections, you see this in the bottom left panel, and dendritic targeting of synapses, which you see illustrated on the right panel. Synapses were placed along the dendrites up to 150 microns away from the soma, but excluding the soma. Note that the left two panels report connection probability and strength values at 75 microns intersomatic distance. But they don't say what the connection probability is as a function of distance between any pair of cells. Therefore, the values from these tables were used as parameters for Gaussian distance-dependent connectivity rules for different source target class pairs. And this is illustrated in the center top panel here. Additionally, excitatory to excitatory connections exhibit like-to-like preference. That is, cells preferring similar stimuli are preferentially connected. So the authors assume that these like-to-like rules are ubiquitous among excitatory connections, and these connection rules are illustrated in the center bottom panel here. These functional rules then modulate the probability of synaptic connection and synaptic strength, a scaling factor between 0 and 1, scaling the connection defined by the other parts in this diagram. The recurrent connectivity parameters need to be fine-tuned to set the right operating condition for the network. The optimization employed a small training set consisting of a gray screen to match spontaneous firing rates and of drifting gratings to match peak firing rates. Epileptic-like activity, where the network would ramp up to have a large global burst of activity and then enter a period of silence until the very next rapid burst is observed for some parameters, but was avoided by the optimization procedure. The optimization uniformly scaled certain selected weight classes, for instance, scaling weights of excitatory layer 4 to excitatory layer 5, all by the same amount as one iteration in the optimization. The authors searched in discrete increments weight changes across connection classes and selected the best result before moving on to the next connection class, although there was still a need to revisit connection classes here and there during this process. Identical criteria were applied to the biophysical and point neuron models. And it turned out that optimizing a full recurrent network at once was very challenging. Instead, the authors followed a stepwise layer-by-layer procedure. They first optimized the recurrent weights within layer 4, here on the bottom left, and then, so with all recurrent connections outside of layer 4 removed. Then they added layer 2-3 recurrent connections and optimized the weights in both layer 4 and layer 2-3, and then step-by-step added layer 5, then layer 6, and finally layer 1. And after optimization, the rates of each individual layer converged to their empirically observed values, illustrated by the vertical black lines in the panel.
Is it okay if I ask a question?
Yeah, please do.
Can you say a few words about I think it's pretty obvious, but could you say a few words about whether this optimization procedure is an entirely artificial optimization procedure that is merely meant to give you the particular tuning and balance that you need for the system to work, or whether the optimization procedure itself is based in some kind of a theory of how the brain does its own tuning to that degree?
So Anton, correct me if I'm wrong, but I believe that at this stage of the optimization, the criteria were only the ones shown on the slide. So spontaneous firing rates, peak firing rates, and the absence of epileptic activity. And direction selectivity and orientation selectivity characteristics were not yet used in the optimization itself. We're going to come to that in some of the upcoming slides.
Yes, Charles, this is correct. And no, we didn't use any learning rules or things like that, or homeostatic rules that brain would probably use. One big reason is that we don't have enough details to do that. Another is that it would be very expensive. So it's sort of an artificial optimization procedure. One thing I do want to mention though is that it was done on a very, very small training set. So it's like a simulation of half a second of gray screen and half a second of a drifting grating in just one direction. And all we ask the model to do is to not be epileptic and approximately get the spontaneous firing rate right and the peak firing rate in response to that single instance of the grating. So it's like a very impoverished training set, and then parameters are fixed, and everything else that's done with many more stimuli is all like test set, if that makes sense.
That makes perfect sense, and the reasons why also make great sense. Thank you.
Thanks.
All right, thanks very much. The optimization turned out to be also very computationally expensive, especially for the biophysical model, but we will come back to that in the discussion at the end. I'll move right along. After optimization, a typical response to a drifting grating exhibits irregular activity with the strongest spiking amongst neurons tuned to that particular grating. So this is matching the firing rates and responses in general of V1 cortex. So after this first training step, the orientation selectivity matches empirical results well. You see the orientation selectivity index plotted in the left panel here for the biophysical model in blue and the point neuron model in yellow. And these are in general corresponding quite well to empirically obtained values. But if you look at the right panel where the direction selectivity index is plotted, the match is a little bit less good. So the authors then further explored the functional rules of recurrent connections, aiming to improve the direction selectivity levels while keeping the firing rates close to experimental values. Maybe one thing also to notice in these figures is that there is really quite a strong similarity between the fully detailed biophysical models and the point neuron models. So these are apparently quite well matched in at least these two indices. And so we may now ask, why do we get such a poor result in the direction selectivity? Well, connectivity probability, as well as connectivity strength, was defined as a symmetric function of orientation alone, and not for direction of motion. So we see the panel here from the previous slide defining the connectivity rules. Connection strengths, as well as the connection probability, is equal for delta theta is 0 and delta theta is 180. This was grounded in empirical data, but actually only the part about connection probability. The strengths were not constrained by anatomical data. Looking at a functional level, similarity to preferred direction alone is actually a poor predictor of synaptic strength, and in vivo patch clamp measurements in layer 4 indicate that excitatory neurons responding to a drifting grading in phase with each other are preferentially connected. So we will now modify the synaptic strength rule in two ways. First, direction of motion-based like-to-like Gaussian profile is applied to all connection classes. So the symmetric nature of this connectivity rule is now altered. And secondly, we're going to confine the sources of connections to a particular neuron to a stripe perpendicular to the target neuron's preferred direction. This will bias the inputs of a neuron to come primarily from neurons that respond in phase with itself when stimulated by a drifting grading or a moving edge. This last rule is illustrated in the bottom two panels here, where we see a schematic of neurons preferring a 0-degree direction as they respond to a 0-degree drifting grading that is shown in the background. Arrow lengths are proportional to phase overlap with the center purple neuron, and the right panel then shows that stronger weights are assigned to neurons that are phase matched with this particular neuron. And then the match of direction selectivity to experimental values is much improved for all cell classes. We see that before, with the symmetric rule, and now with these modifications on the right, we get a good match between model and experiment. However, these new connection rules introduce a firing rate bias depending on the neuron's preferred direction of motion. Vertical preferring neurons exhibit higher peak firing rates than do horizontal preferring neurons. But such a bias is not present in empirical data. The root cause of this is the experimentally observed asymmetric retina-topic mapping in cortex, which, as we mentioned before, is implemented in this model. So 10 degrees of movement in the visual space corresponds to 250 millimeters of movement in the cortex for elevation, but only 140 for azimuth. So the stripe that we saw in the heat map of the previous connectivity rule is actually wider in cortical space for vertical than for horizontal preferring neurons. So this provides a stronger net input from presynaptic V1 neurons for this particular orientation. So because this firing rate bias is not empirically observed, you can see this in the left panel, some mechanism must adjust for this horizontal-vertical mismatch of translating retinotopy to connectivity. For instance, some adjustments that we could implement is a different distance dependence of connectivity rules for vertical versus horizontal preferring neurons, different strengths of LGN inputs for different orientations, or different strengths of recurrent connections. The authors implement the latter, so different strength of recurrent connections, in a simple linear fashion where horizontal preferring target neurons are have synapses that are scaled up by a factor of about 1.4, and vertical neurons have synapses that are scaled down by a factor of about 0.8, with a simple linear interpolation in between. In that final model, horizontal and vertical preferring cells then receive on average equal amounts of excitatory synaptic input, sourced from the same size of stripes in retinatopic space, but different sizes in physical cortical space. Consequence of this, you see in the figure on the right, is that the distribution of incoming excitatory weights for a particular neuron has a heavier tail for the horizontal than for the vertical preferring neurons. An observation that could be tested in future experimental data sets as an indication of the mechanism implemented here. So now the model is making a prediction that can be empirically validated. With the final synaptic design in place and having observed good model performance for drifting gratings, the authors model responses to full field flashes, so at full field luminance change. This is one of the strongest stimuli to test the stability of the network, as well as response latency. Despite the highly correlated structure of the input, individual neurons tend to have low correlations with each other in both models, in the biophysical and the point neuron model, as well as in the experimental data, as you see in the figure on the bottom right. The signal correlations in the models overlap closely with the experimental ones. And in the top right panel, you see the time to peak values for responses to these full field flashes. And it is found that these are indeed comparable to the NeuroPixels experimental recordings. This is an important indication that the dynamics of this initial transformation of the visual signal is captured relatively well. Then the authors test their models on a clip from a movie shown to mice also empirically in the Allen Brain Observatory, which use calcium imaging to quantify these responses and allows a comparison between experiment and model. An example raster plot shown in the middle highlights more irregular and sparser responses than responses to drifting gratings or full field flashes. Again, the correlation between the signal correlation and the noise correlations for neuron pairs is analyzed and compared with experiment. And in the top right panel, you see that indeed there is a good match between the model and experiment. And then finally, the bottom right panel shows the lifetime and population sparsity of neurons. And again, a good match is observed. So to summarize, we have seen a data-driven view on model, allowing arbitrary visual stimuli like movies to be simulated and neuronal responses to be recorded. The model aims to not just replicate one or two aspects of V1 behavior, but to reproduce in a single model and from a single set of parameters many V1 phenomena at a time, such as orientation, spatial frequency selectivity, spontaneous and driven firing rates, gamma rhythms, simple versus complex cells, etc. This study demonstrates that it is in principle possible to reconstruct this integrated view of the structure and function of neocortical microcircuitry, using sparse complementary data sets to predict biological parameters that have not been measured experimentally. The model makes predictions, so we've seen in particular these two predictions about how synaptic weight rules could be substantially different from synaptic connection probability rules, so the probability of connection can be different from what the strength of that connection would then be. And the second prediction that the model makes is that there is this asymmetry due to the cortical retinotopic mapping. There's in general very good agreement between the biophysical and GLIF models using the metrics used in the paper. Although the connectivity graphs are identical, the input-output function of every neuron is very different. But judging by their firing rate distributions, the two models act similar at the population level. And in the words of the authors, this reveals, yet again, the perhaps unreasonable effectiveness of point neuron models, given their vastly reduced degrees of freedom. In general, the level of resolution to use in a model must be based on the scientific question under investigation. For instance, computing the extracellular field potential requires spatially extended neurons. Passive dendrites were used in the model. This was due to active dendrite models being too computationally expensive for the extent of the work, especially when using that in an optimization loop. And the the GLIF models are the point neuron models, which were about 8,000 times faster to simulate. This is really quite a radical speed up. Just one word about the software infrastructure. So absolutely my hat off to the fact that all of the data is being made available through these various resources in open data formats, and that also the computational model itself is being made available for download in an easy to reproduce manner, so using virtual machines or Docker containers. And this allows others to very quickly reproduce the results in this paper and perhaps more importantly, to build further upon this work. This model is part of an overall trend towards increasingly sophisticated models of cortical circuits in general. Some of these model a specific sensory modality, or are models of cortex in a more general sense, but especially layered models of single cortical areas, as well as multi-area models. An overview table of some models is provided here on the right, where there is some comparison on the basis of phenomena reproduced by the models. Another comparison diagram I took from a PhD thesis here. Unfortunately, this PhD thesis was published before the paper that we're discussing, but I think that the current paper would fit somewhere in the middle of this diagram. So I'd love to get into a discussion about the details of this work. Maybe here are just a few points to get us started. So the level of detail required, so there was a good match between the two variants of the model, and this was quantified by the so-called similarity score based on a Kolmogorov-Smirnov test that calculates the distance between two probability distributions and is bounded in the range 0 to 1. Unfortunately, I found this still a little bit hard to interpret. Like, what does it mean for a similarity to be 0.88? Software infrastructure, as we've seen in the previous slide, is very open. So this is really a big advantage, especially as these models are becoming more integrated. And if you want to integrate one other biophysical characteristic, you don't have to reinvent everything from scratch, but you can build upon existing work. In the model, many parameters are optimized on the basis of a computational interpretation of connectivity. So we were reasoning already on a computational level about orientation selectivity and in particular direction selectivity, but these notions of computation might not in general be available for all parts of cortex. Then a final point, parameters that were directly based on in vivo data were not allowed to be optimized. So if a parameter was obtained from the literature, then in the optimization it was fixed or only allowed to be optimized by a factor of 2, so to be reduced a factor of 2 or incremented by a factor of 2. Now, hypothetically, this is looking a little bit into the future. What if we begin to fix more and more parameters as more empirical data become known, and at some point the network stops working or it stops behaving the way we want it to behave? This would then probably be an indication that we're still missing some other crucial mechanism, but in the face of a mechanism still missing, should we actually maybe allow more robustness in the network by allowing all parameters in principle to vary? Then my final slide, I thought I should say a few words about this, how this work relates to the Aspirational Neuroscience Award. The challenge is to decode a non-trivial memory or function from a static map of synaptic connectivity. As we've seen, the connections in this model were based on statistically defined synaptic connectivity. So we had a certain probability distribution of the probability of connection and the strength of a connection, and not on an actual map of a particular mouse's viewer connectome. But it seems clear that the modeling and analysis tools developed here are geared towards that eventual goal. The computational requirements would seem equivalent in terms of the number of neurons and synapses that have to be simulated. So this work is coming from the primarily from the LDN Institute for Brain Science, the same institute that has recently imaged the cubic millimeter volume of mouse visual cortex. So we certainly hope that there will be some cool follow-ups to this already very cool work where the connectivity is based on electron microscopy reconstructions. That was my presentation. Thank you very much, and look forward to further discussion.